In the genetics department at the California Institute of Technology, Pasadena, the breeding room, where fruit flies are kept at the optimum degrees of temperature and humidity for reproduction and development. The jars contain living material for dozens of genetic experiments. We are going to carry out a simple but important experiment producing mutant fruit flies by exposure to X radiation. These are wild-type flies of the species Drosophila melanogaster. They're etherized, then the males are sorted out. Males have, as well as other distinguishing features, black tips to their abdomens. Groups of 30 males at a time are placed in small glass tubes. The effect of the ether will soon wear off, and the insects are not harmed by the small dosage they've been given. Fruit flies make excellent subjects for genetic experiments, since they are easily managed and have quite short reproductive cycles. The male Drosophila are now taken for exposure to X radiation. X-rays can speed up the rate of mutation of living things. Mutation involves affecting the genes so that progeny have changed characteristics of one kind or another, which they in turn pass on to their offspring. They are inherited changes. Mutations occur naturally. Radiation, such as X-rays, simply speeds up the rate at which they occur. The same mutations as in nature, but turning up more frequently. The fruit flies will now receive a measured dosage of X-rays. The heavy door is part of the shielding essential to protect the operator from possible exposure to the radiation. Radiation is now beamed onto the flies, and this will cause changes to the genes in the sex cells of at least some of these males. The irradiated males will be used in breeding experiments. Any mutations caused by the X radiation will produce changes in their offspring. The males are placed in jars containing normal females with water and a food supply. Each tube of 30 males is shaken into a different jar of females. The males will mate with the females, and the females will lay eggs. These hatch to produce larvae. The larvae pupate, and then adult flies emerge from the pupae. One cycle takes about 12 days in the warm, humid atmosphere of the breeding room. The offspring of the irradiated males can now be examined. But first of all, let's look at normal specimens of Drosophila melanogaster. This is the normal female. Note the red eye. And the normal male with black-tipped abdomen and red eyes again. Another useful thing about Drosophila is that many of the mutations which it can undergo are easily spotted. Here's one. This male, in a line descended from one of our irradiated males, has white instead of red eyes. This one shows another mutation. It's called bar-eyed and has slit-like red eyes. This third mutation has shorter wings than normal. Look where the wingtips are in relation to the end of the abdomen, and compare it with the normal male, whose wingtips project well beyond the end of the abdomen. We can sort out these mutant males, which are, remember, the descendants of our original irradiated males, and mate them, each mutant type, with normal females. What descendants arise from the white-eyed males mated with normal females? Well, they give rise to further lines of descent with white-eyed males. You can see the white eyes on the 5 males at the top of the picture. The change produced in the original males by the X radiation is an inherited change, a gene change, a mutation, producing a variety which breeds true in the male line. What about the bar-eyed mutation? Is this passed on by inheritance? Yes. Once again, some male descendants turn out to be bar-eyed. Look carefully at the 6 males at the top, and several of them are in a good position to show their slit eyes. The male mutants with miniature wings, do they pass on this variation? Yes, again the males with the black abdomens in the top half of the picture all have the short wings characteristic of this mutation. Compare the longer wings of the females below. What the X-rays have done is to speed up a process which, as we've said, can also happen in nature, the sudden appearance of new varieties, which must be caused by some kind of change in the parent, transmitted to its offspring and to all future generations. Another experiment here at Caltech demonstrates the production of mutations, not using radiation this time, but a chemical substance which causes mutations, a chemical mutagen. This experiment also makes it clear that it is the mutagen which causes genetic change. Many chemical substances can act as mutagens. This one, ethidium bromide, is particularly powerful and must be handled with great care. Van Parker, a research student, adds ethidium bromide to a nutrient agar which also contains an antibiotic. He makes up 3 samples containing respectively little ethidium bromide, a medium concentration of ethidium bromide, and a high concentration of the mutagen. There's also a control containing no mutagen. This tube contains a culture of the bacterium Salmonella. Using the sterile techniques essential both for success and for safety in all microbiological work, portions of this bacterial broth are added to each of the 4 agar preparations. The antibiotic present should kill off the bacteria, unless there are any mutant bacteria present which have antibiotic resistance. Here's the final tube, the control, containing just agar and antibiotic with no mutagen added. In goes the Salmonella. All 4 tubes have been kept warm so that the agar doesn't yet set to a gel. Plates are now prepared. The tubes, containing agar, salmonella, antibiotic, and in all except the control different concentrations of ethidium bromide, are poured into separate sterile dishes. If ethidium bromide acts as a mutagen and causes salmonella to become antibiotic resistant, the bacteria will multiply on the plates, and we might expect more mutants to be produced the more concentrated the mutagen. Here's the final sample containing no ethidium bromide. The plates are taken to an incubator and left to develop. Here's the control. There must have been a few antibiotic-resistant bacteria present in the original population. These have survived and indeed multiplied to produce about 20 visible colonies. Untreated salmonella contains these resistant bacteria produced by natural mutation and part of the normal range of variation. Now for the plate to which dilute ethidium bromide has been added. there are just a few more colonies, some of them small, suggesting that the ethidium bromide may have induced some extra mutations, producing a slightly greater number of antibiotic-resistant bacteria which have multiplied to produce these colonies. A greater concentration of ethidium bromide has had a much greater effect. There are far more colonies, showing that the higher concentration has increased the frequency of mutations. What we're seeing, of course, is the selection of a particular mutation, antibiotic resistance, by this antibiotic-contaminated environment. Finally, with the greatest concentration of the mutagen, a very large number of mutations have been induced, and the plate is covered by the colonies produced. Clearly, the chemical mutagen acts in some way on Salmonella, changing its genetic potential so that it produces antibiotic-resistant offspring. The more of the mutagen, the greater the effect. When mutations occur, whether in Salmonella or those fruit flies we saw earlier, or indeed in any living organism, some set of instructions within the organism which governs the characteristics of its offspring must be changed. How are the genetic instructions carried inside living cells? In one of these buildings in New York, at what's now the Rockefeller University, one of the most important biological discoveries of all time was announced in 1944 by three scientists, Avery, McLeod and McCarty. They discovered what was called the transforming principle. Dr. McCarty explains.
The Avery Laboratory at the Rockefeller Institute for Medical Research had been concerned for years with the problem of pneumonia. The primary cause of pneumonia is a bacterium which is generally called the pneumococcus. The work in that laboratory had been principally responsible for showing that the pneumococcus is made up of several different types, which were just given numbers, and the different types were dependent upon different capsules which surrounded the bacterium. The capsules were chemically and immunologically distinct in the different types. The importance of the capsule in terms of the study of the disease was that it is responsible for the protection of the organism from the defense mechanisms of the host. That is, it protects the organism from being phagocyted and destroyed by white blood cells.
Many types of pneumococci have quite thin capsules or no capsule at all. We can represent one of them like this. When it divides, it produces similar pneumococci with the same thin capsules. But certain pneumococci have thicker capsules like this. When these divide, their daughter cells also have thick walls. This makes it difficult for the body's defenses to penetrate and destroy them, so they constitute, medically, a great danger.
It was shown that one could make extracts of one type of pneumococcus and use that extract to transform another pneumococcus to the type that the extract had been obtained from.
It was discovered in the Rockefeller laboratories that if material was taken from the thick capsule pneumococci and transferred to the thin-walled type, the thin-walled pneumococci then divided to produce the thick-walled variety. The descendants of the originally thin-walled bacteria were all thick-walled. They'd been transformed by some substance, the transforming principle, originally present in the pneumococci with thick walls.
So it was this pattern of transformation of pneumococcus with material extracted from the bacterial cell that set the scene for the later studies in Avery's group to find out the nature of the substance in these extracts that caused this transformation. The reason for the interest is fairly obvious because the material in the extract was changing a living cell in a way that it did something it hadn't done before, made a product that was new, and continued to make this product after that in continuous culture. This suggests some kind of transfer of a hereditary principle. And it was for this reason that the efforts were directed toward trying to determine the nature of the substance in extracts of pneumococci that would cause this change. There was no basis for prejudging what this material might be, so the kind of studies that had to be done were fractionation and attempting by various means to eliminate one component or another. And it was out of several years of this kind of analysis that the conclusion was drawn finally that the material that was responsible for the transformation of pneumococci was desoxyribonucleic acid. This came as something of a surprise because of two reasons. One being that it had been assumed quite widely that the only substance in nature that had a sufficient degree of specificity to carry genetic information would be protein. This was thus the first experimental evidence that DNA carries hereditary or genetic information.
Let's see how the DNA, deoxy or desoxyribonucleic acid, can be isolated from this fresh cast liver in the laboratory. It's homogenized in buffer solutions. The solid detritus is filtered off, and the filtrate is shaken with phenol and chloroform, which denatures and absorbs protein, leaving DNA in the aqueous buffer solution. This is then centrifuged. A short run at 1200 revolutions per minute spins down solid matter. The supernatant liquid now contains the DNA in solution. It's carefully pipetted off. Now alcohol is very carefully added so that it forms a top layer. At the interface with the alcohol, DNA is precipitated. It comes out as fluffy white filaments. This is the chemical substance which carries genetic information inside the cells of a living organism, in this case a calf. DNA from other living things looks just like this, but carries, for every organism, the special genetic instructions unique to that individual, whether it be a plant or an animal, a bacterium or a man. Chemical investigation of DNA has shown us its highly complicated structure, long-chain molecules containing many thousands of atoms, of which this model only represents a small portion. In the next film, we shall see how DNA functions and what part it plays in producing the great diversity of living organisms on Earth today.